Okay, so now that we know about the different cell types and about the phospholipid bilayer, we need to look at organelles inside of different plants and animal cells. First of all, we'll start with the nucleus. And the nucleus is what contains the chromatin. Chromatin is both the DNA and the associated proteins. And we'll talk some more about chromatin in a later chapter, so we'll learn about its components. But the nucleus is kind of the control center of the cell. That's what sends out all of the information on what to build and what to do and how to function as a cell. Next organelle is the cytoskeleton, and the cytoskeleton is a fairly diverse group of different filaments made up of protein inside the cell. It gives shape and support to the cells. It also helps to move stuff around, both inside the cell, intracellular, and move the cell itself. So for shape and support, it just forms a network of fibers that connect to organelles and hold everything kind of in place and keeps the shape. It's so dense, in fact, that if you take a cell and digest away all of the internal organelles, leaving the cytoskeleton in place, you can see right here is where the nucleus was in this cell. So you can actually see where different organelles were based on what's left of the cytoskeleton. For intracellular traffic, all of these fibers and filaments can work like a monorail system. So there are motor proteins that can actually attach to these and zip along them like a monorail to get across the cell or to different points inside the cell. And that's how your cells actually move or transport things around internally. Movement, as far as external movement, some cells have cilia and some have flagella. The main difference between those is their number and their length. So cilia, there's going to be lots of them and they'll be much shorter. Flagella, there'll be just a few, and they'll be much longer in structure. These show the cilia lining your lungs, so when you breathe things in, so dust, dirt, pollen, other things, these cilia around the air sacs in your lungs will actually grab all of that debris and start passing it back up and out your throat. You either swallow it and it gets digested, or it comes all the way up and you spit and hack it out or blow it out your nose, and that keeps your lungs clean. If you're a smoker or if you're around smokers on a regular basis, the different chemical components inside cigarette smoke will paralyze these cilia, and that's what causes the smoker's lung effect. So your lungs start accumulating not only the tar and residue from the cigarettes, but also from just the environment. So pollen and dust and all of that gets stuck down there because the cilia are not working properly. Flagella, these look like sperm cells, and they just have one long flagella, allowing them to swim and move. Also, there are flagella in the reproductive tract of females. So if you look at a female reproductive tract, here's the fallopian tubes on either side. At the end of the fallopian tubes, there's the ovaries, which actually produce the eggs. There's a gap between where the eggs are produced and where they go into the fallopian tubes, and this just drops out into the body cavity. So to catch all of the eggs as they're produced, there are cells inside the fallopian tubes that have flagella anchored to them, and those flagella move in unison and create a current which pulls the egg into the fallopian tube and down into the reproductive tract. Usually. Sometimes the eggs miss and actually fall out into the body cavity. That's very, very rare. Sometimes it happens. Mitochondria are the next organelle, and mitochondria produce all the energy for your body. So you eat your food and it gets digested in your stomach, goes to your small intestine and broken down further and absorbed into your bloodstream, and then that energy is picked up by cells that take your food molecules, the sugars, and pass them to the mitochondria. The mitochondria actually breaks that sugar down and produces something called ATP, which is what the rest of your cells in your body use for actual energy. So this does energy conversion. And there's different numbers of mitochondria depending on the type of cell. So red blood cells have 0 functioning mitochondria, all the way up to things like liver cells that have 2,500 functioning mitochondria per cell. Skeletal muscles, just over 1,000. And you can actually increase the amount of mitochondria in things like your skeletal muscles. If you've ever done endurance training, so if you're an athlete and your coach makes you do endurance training, that's not real fun. Basically, that pushes you right to your limit and a little bit beyond so that you're utterly exhausted by the end. The reason you do that endurance training over a period of time is that tells your cells, hey, we're running out of energy, we need more, and your cells actually start producing more mitochondria in each of your skeletal muscles. So that allows you to turn energy over faster and for longer periods of time because you have more mitochondria in those cells. So that's what endurance training does for you. Also, mitochondria have their own DNA, and this DNA is really interesting because it's circular DNA. And if you remember when we talked about different types of cells, in prokaryotic cells, those bacterial cells, there was that circular DNA. And we looked at how mitochondria was probably at one time a separate living organism because of that circular DNA, also because of this double membrane structure and several other components of mitochondria. Something else very interesting about mitochondrial DNA is because it's small and it's a high copy number, basically each mitochondria has hundreds of these tiny little circles of DNA, they can last for even longer periods of time than the DNA in the nucleus. So they're much easier for researchers to get at and sequence. And so then you can look at kind of the history or the DNA in the mitochondria. Mitochondria only pass from the mother, so all of your mitochondria always comes from the mother, and females pass all their mitochondria to their offspring. So you can look at kind of descendants on the maternal line using mitochondria. One kind of sad example where that occurred was with Anastasia. And you guys are possibly familiar with the Disney movie. So in the Disney cartoon, here's Anastasia. Here's a con artist she eventually runs off with. Her grandma would be proud. And this fellow over here is Rasputin, and he's rotting and falling apart, and he's got a little friendly bat, which helps him. And Rasputin's the bad guy, and he's, trying to get Anastasia. Well, the real story of Anastasia is not really fit for a Disney movie, so we'll talk a little bit about the real family and Anastasia and how mitochondrial DNA was used to solve the mystery of Anastasia. So here's the Romanov family. They were actually the ruling family of Russia, so kind of the Tsar or Emperor of the entire country. And so here's the Tsar and his wife and their children. So they had mostly daughters, and this one's Anastasia. They did have one little boy. I think his name was Alexei. And Alexei, this little boy, had hemophilia. And hemophilia is a blood disorder where your blood doesn't clot. So you could die from bleeding from a paper cut. You could die if you got a bruise, because a bruise is just bleeding underneath the skin. So a bruise could actually kill you because you just bleed out underneath the skin. So Alexei here was a very sick young man. And what had happened is this Romanov family had called lots of different doctors, different people in to try to help with their very sick son. And one of the people that became very close with the family was a guy named Rasputin. Politically, he was very unpopular, but he had been called in once and he prayed and did something over the boy, and he recovered. And so they put a lot of faith in this Rasputin fellow, and he had access to the family basically anytime he wanted, and people were concerned about Rasputin's influence on them. So concerned, in fact, that they murdered Rasputin, so he was never around to be the bad guy, as depicted in the Disney movie. But soon after the murder of Rasputin, there was a full government overthrow, and they took the entire Romanov family along with kind of close associates. So there was a doctor with them at the time, and kind of their maids and immediate people associated with them. They took that whole group and marched them out to a house in the country, and they took over the government. Now, they had had this family out there, and they actually moved them around a couple of times, but they'd had this family basically in prison for a while, and they were now running the government, this group that overthrew them, and they decided, well, some people really liked the Romanovs, so it's kind of inconvenient to have them around. They were worried that they would come back and overthrow the overthrowers. So the family just disappeared, and it was not known for a long period of time, about 10 years, what actually happened to them. They just vanished. What had happened is a group of executioners had been sent out to a little farmhouse in the country where the family and their associates were being held, and they were all assassinated. So that happened on July 17th of 1918. So Nicholas, his immediate family, and all the people with him were all assassinated. This was learned or discovered because one of the assassins finally came forward and said, well, yeah, they sent us out, we marched them down to the basement, we lined them all up and basically did a firing squad, shot them all to death, and then, because nobody had ever found any bodies, the executioner let them know where the bodies were, said that they'd thrown them all down a well. So they went and excavated in the well, and they recovered the remains, and were able to identify all of them, except they were still missing the two youngest children. So they went back to the executioner and said, okay, thank you, we found everybody except we didn't find Anastasia or her little brother Alexei. Where did those children go? And the executioner at that point said, oh yes, I remember now. We burned their bodies in the woods right over there. So they went to the woods and they again did some excavating, and they did find bone fragments that they identified as the little boy. So he had been found. However, they didn't find any remains of the little girl, Anastasia. And again, this is about 10 years later. So this is when the rumors surfaced that, well, maybe Anastasia had survived because they couldn't find her. No remains were found at the time. Since then, they've found some. But at the time, they couldn't find any remains from this little girl. And so in a neighboring country, a young woman had been brought into a hospital, and she had tried to commit suicide, I believe, by drowning herself. She was brought into the hospital, and when she woke up, she couldn't remember who she was. Didn't know her name, why she tried to do that. Couldn't remember anything about herself. And the nurses and people at this hospital were looking at this young woman and said, well, she kind of looks like a Romanov. She's got the same body type, same eyes. Aren't those the Romanov eyes? And so there's the rumor that perhaps she's still out there. And there's this young woman about the same age, about the same physical description that shows up in a neighboring country in the hospital. Can't remember who she is. So perhaps this is Anastasia. She just has amnesia. So she actually was supported by a lot of people for the rest of her life, people believing that she was this missing Anastasia. She ended up marrying a doctor, and I think she lived on the east coast of the U.S. until she was an old lady, and she died naturally. After her death, however, there was a huge court battle over an envelope of hair she'd left behind. She'd pulled the hair out by the roots, and so it had that little skin tag at the bottom, and there's DNA in that skin at the base of your hair. So in the court battle, people who wanted to have it tested to prove if she was or if she wasn't related to the Romanovs, and people who didn't. The people who wanted the testing won, so they were able to send her hair for DNA analysis, and they compared that to the maternal line, because remember, mitochondria comes from the mom. Compared that to the maternal line and found that it was not a match. There was no possible way that this woman had been the missing Anastasia. The little girl probably died along with her family at the time of the execution. However, they extended their DNA search and found that there was a family of farmers in the area where this woman was found whose daughter had vanished, gone missing at the same time she was found in the hospital, and she was a perfect match for that family. So I suppose if I thought things were bad enough to try to commit suicide, and I woke up in a hospital and everybody believed that I was a missing princess, I would definitely have amnesia too. Alright, so our next organelle is the lysosomes, and lysosomes inside the cell, they're responsible for recycling. So they break down, you can see this one's breaking down kind of mitochondria that's no longer functioning correctly, so they dissolve and digest anything that's kind of old or not functioning correctly, and then they allow those components just to be recycled and reused. So it's filled with very, very powerful digestive enzymes, and these are really important during development as well. If you look at your fingers and toes, now in class I'd have you hold your hands up. I don't want to see your feet, but we'd hold our hands up. If you look between your fingers and toes, do you have any webbing between your fingers and toes? If not, you can thank lysosomes. So during development, have you ever seen pictures of babies during development? Early on, their hands are kind of paddle-shaped. You can see the fingers, but they're all connected by a flap of skin between all the fingers. Well, during development, that skin forms, and then later on, they get a signal from the body or from the DNA that says, hey, we don't need that skin anymore. We're not going to develop into a webbed-fingered creature. So it sends a message to the lysosomes to break open, and when they break open inside those cells between your fingers, it just digests away that skin. So that skin basically disappears between all of your fingers and toes, which is why you don't have webbed hands or feet. Or if you were born with webbed hands and feet, mommy had that surgically removed, and we just don't talk about your webbing. So that's what lysosomes do. The endomembrane system, it's a series of basically sacs that are formed with membrane, and we'll look at the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and the Golgi apparatus. Structurally, they're all very similar. You can see here's the rough, here's the smooth, and here's the Golgi, and they're all just kind of flattened sacs of membrane, but they all have different, functions in the cell, and we'll look at each of those. So first of all, the rough ER. The rough ER is covered with ribosomes, these little purple dots all over. Those are ribosomes. And you've seen ribosomes before. When we looked at our prokaryotic cell, we had DNA in there, we had little circles of DNA, and we had all those dots, those purple dots. And those were ribosomes in prokaryotes, and they also made proteins in the prokaryotic cells. So in eukaryotes, on the surface of that rough ER, it's covered with ribosomes, so it makes protein. So it produces protein, and it helps those proteins to fold properly so that they function. The smooth ER looks slightly different, but again, just formed of kind of flattened membrane sacs. The smooth ER produces lipids, so makes more fats and lipids, like more phospholipids that would be exported to make more membrane. It also helps break down different toxins in the body. So it's a smooth ER that helps metabolize or break down alcohol, antibiotics, barbiturates, amphetamines. Some of these you should not put in your body. But over time, the smooth ER can do very much like the mitochondria. So remember when we talked about the mitochondria, endurance training can increase the amount of mitochondria. Well, if you drink alcohol on a regular basis or take antibiotics or any of these other illegal substances, then you're going to force your cells to respond by growing more smooth endoplasmic reticulum. And as you have more and more of the smooth ER, you need more and more alcohol or antibiotics to get the same, effect. The next part of this endomembrane system is the Golgi apparatus, and the Golgi apparatus will add stuff to proteins and do modifications. So we've talked about carbohydrates or sugars that help identify your red blood cells, things like that. It's inside this Golgi apparatus where those sugars are added to the proteins before they're exported. So they do any modifications to proteins or other molecules, and they can tag things for export outside of the cell or to go anywhere inside the cell. Looking at the whole endomembrane system together, again, we have our rough ER, which makes proteins, and they can be passed to our Golgi apparatus through vesicles. Anything that's just a little circle of membrane being transported, that's a vesicle. Our smooth ER, again, makes lipids and also breaks down toxins. It can make things and transport them to our Golgi apparatus. The Golgi just helps package, tag, and modify, and can export them outside the cell or to any other area inside the cell. we'll look at a couple of organelles that are only found in plants. The first is the plant cell wall. And the cell wall is this huge, wide structure here surrounding every plant cell. You can actually see that in this microscope slide here. All of these are the cell walls. And plant cell walls are what give plants their strength. So some of them are very strong. The strength of this entire tree that this poor guy's climbing on, the strength of that tree, that's just the combined strength of each individual cell and the cell wall of each of those. And cell walls are composed of something called cellulose. Cellulose is pure sugar. It's glucose units linked together. The problem with cellulose is we can't digest it. We don't have an enzyme capable of breaking apart that particular bond that's in cellulose. So if we eat cellulose like grass, it comes out as, well, grass. It doesn't get digested or broken down. Things like bacteria, certain types of bacteria, can break apart those glucose units and actually get sugar out of cellulose. So any organism that eats cellulose as a main dietary staple is going to have a relationship with bacteria so that they can digest it. A couple of examples, termites. Termites eat wood. That's just made up of cellulose. They can only digest it because inside the guts of termites, they are hosts to the type of bacteria that can break cellulose. So they eat the cellulose, the bacteria actually digests it for them, and they absorb the sugar. Another thing, like cows. We're in Iowa, so maybe you've seen cows standing out in a field, and cows, they eat grass, right? Cellulose. And in order to digest that, they're ruminants, so their stomachs have multiple chambers. And what they'll do is they'll chew the grass for a while, and they'll swallow it down, and it will roll around in their stomach, which also contains a similar type of bacteria that can break cellulose down into sugars. So it'll roll around their stomach for a while with the bacteria, and then cows do something that's called chewing their cud. Basically that means this little ball of grass that they've swallowed and partially digested, they regurgitate that, so puke it back out into their mouths, and they chew on it for a while longer. So they'll stand there and chew and chew and chew, and that just helps mechanically rip apart the grass so that the bacteria can get in there to all of those cellulose links and break it apart. And then they'll swallow it again and puke it again and swallow it again until eventually they've extracted as much energy as they can out of that grass. Another structure that's usually found in plants, there are a few animal cells that have these, but we'll stick with that it's usually in plants, are vacuoles. And vacuoles work for storage. So they're just large sacs inside of plant cells. So they can store water, they can store waste, they can store toxins that are used in defense. Some plants use them for reproduction. A lot of plants use them for support. So a lot of soft plants, so not woody plants like trees, but soft plants, they will absorb water and fill their vacuoles, and that will pressurize each cell so they can stand upright. And it will start to fall over and get limp as they lose water and those vacuoles empty out. So vacuoles are found primarily in plants. Chloroplasts are found again only in plants, and these produce energy. So these can use sunlight to produce energy. Basically, wavelengths of sun will strike these thylakoids inside the chloroplast, and they'll use that to generate energy, ATP. Interesting thing about chloroplasts is very much like mitochondria. They have their own DNA and similar characteristics to mitochondria, which shows that these were also a separate living organism that migrated into plants not just once, but multiple times, because there's different types of chloroplasts out there. Here's a review of all the structures. So notice most of them are found in both animals and in plants. So the nucleus, the cytoskeleton, that's in both. The mitochondria, that is in both. Plants and animals have mitochondria. The lysosomes are in both. The rough, the smooth, and the Golgi are in both. Cell wall, only in plants. You don't find the cell wall in animals, so that's a main difference. The vacuole is sometimes found in animals, although mostly only in plants, and the chloroplasts are only found in plants. So if you remember at the beginning of this chapter, I let you know that one of my favorite exam questions is to put a diagram of either a plant or an animal cell on the exam and ask you, well, is it a plant? Is it an animal? And how do you know? You would know based on these three structures, even though it's sometimes, we'll say for this class, vacuole is a plant structure. So that way you have three structures that are found only in plants. So if I give you an animal cell, will you tell me it doesn't have a cell wall, it doesn't have a vacuole, and there are no chloroplasts, so it must be an animal. If I give you a plant cell, you can tell me, well, it's got a cell wall, vacuole, and chloroplast, so it must be a plant. So just be able to recognize those differences. To help you with recognizing plants and animal cells, and also just to become familiar with the organelles, your assignment, again, due next time you come to class, is to make a diagram of a plant and an animal cell. So describe in your own words, so don't just copy out of the book, in your own words, describe all of these organelles. I starred the ones that are only in plants, so describe all of these just on a sheet of notebook paper or type it up if you're worried about me being able to read your handwriting. Describe them, and then separately draw a plant and an animal cell. If you're just drawing them, make sure you use a sheet of paper for each and draw the cell to take up the whole page. So I don't want little miniature drawings, just draw them nice and big on plain paper, not lined paper, and then write your descriptions separately. This assignment, several assignments through the semester, you can get extra credit on the assignment if you do an exceptional job and I see that you've spent some time and thought doing it. To get extra credit on this assignment, be creative. Whatever you'd like to do, there's nothing specific that you need to do. So a very easy way to do this is to take your time drawing the cell. Use colored pencils or crayons and color in your different organelles. That's going to take you more time and effort than just a quick sketch on a sheet of paper before you run into class. So use colors, be creative. Students in the past, they've made books. So the organelles were all characters in the book, and the characters have, each of the organelle functions, and they interact to do something in the cells. That's an example of something you can try. Other students, it's very common to draw the cell and as a city. So each of the organelles represents different kind of parts or buildings or structures in a city. So you might have City Hall as the nucleus, that's the command center for the city. Then you'd have a manufacturing plant as your rough ER, making things. Your Golgi apparatus would be like UPS or some delivery service, transferring stuff around the cell or outside of the cell. So just be creative, whatever you'd like to do. An example of something else that a student has done with this assignment is drew that as an animal and as a plant and tucked all the organelles inside of there. Again, be creative and do not copy anybody else's design. Try to make your own cells in a creative way. The point of this activity is to help you remember what those cell structures are. So think about what might help you remember the different organelles in an animal cell and in a plant cell and draw it. If that's just copying out of the book what they have drawn and coloring it in, that's fine. You can still get extra credit. But if something more creative would help you, then please do that.